Okay, so this is chapter 1, which is titled Biology, the Study of Life. And hopefully by now you have read this chapter in the textbook, and also hopefully you have completed both the vocab list as well as the guided chapter outline. Okay, and so as you watch this presentation, you want to make sure that you have your PowerPoint slides available so that you can take notes on them. Those are on Blackboard for you. You want to take notes throughout the lecture presentation. And every 10, sometimes 5, sometimes a little more than 10 slides, we're going to take a pause and you are going to answer some of the "Are you with me?" questions. So there's another assignment on that list, right? So you have your vocab list, you have your guided chapter outline, that's followed by the PowerPoint presentation and those slides, and then underneath that, the other assignment is going to be the in-class assessment, or the "Are you with me?" So you want to make sure that you have that in front of you as well, so that when we get to those pause points, you can go ahead and answer those questions. And remember, you can answer those electronically, or my preference would be for you to print out those pages and to handwrite your answers on there. There's lots of studies that show that handwriting, vocabulary or handwriting answers to questions helps you to remember the terms just better than doing it by typing. Okay, you'll also see, I'm not sure in chapter 1, but in future chapters, some of those "Are you with me?" questions ask you to draw things or sketch things. And so again, doing it on paper might be a little bit easier for you. Okay, and then you can scan those documents or take pictures of those documents with your phone, and then you can upload them right to Blackboard. Okay? Okay, so let's get started. So again, this is chapter 1, the study of life. So here's a picture right from your textbook. Obviously, this is Earth. Our planet is about 4.6 billion years old. And we believe that life arose on the planet about 3.8 billion years ago. So life has existed on the planet for a pretty long time. And we have evidence of this in our fossil record. Okay? The first organisms that evolved on the planet were going to be microscopic, very, very small, single-celled organisms. We're going to go over cells in a couple of chapters. All living things are made of cells. You're made of trillions of cells. The first life on the planet was probably made of just one single cell. And that single-celled organism was most likely a prokaryote. So you can see that bold-faced term there, prokaryotic. I'll underline that for us. Okay? The term prokaryotic means before a nucleus. Okay? So kary means nucleus and pro means before. So before the nucleus. So these single-celled organisms, they did have genetic information, so they did have DNA, but that DNA was not housed or bound in a membrane-bound compartment. So they are prokaryotic. They do have genetic material, but they do not have a nucleus or other membrane-bound organelles. So those are probably the first organisms to appear on the planet. And then about 2.7 billion years ago, eukaryotic organisms appeared. Okay, and those were probably started off as single-celled, and then multicellularity did evolve from there. That term eukaryotic is the opposite of prokaryotic. So eukaryotic organisms, they have their genetic material, their DNA, housed in a membrane-bound compartment, protected. Eukaryotic organisms also have other organelles. These are tiny organs of the cell. So we're going to learn all about these in chapter 4, but just to give you an idea of what some of those might be, you're probably familiar with some of those. If you've heard of mitochondria or chloroplasts, or the Golgi apparatus, or the endoplasmic reticulum, vacuoles, vesicles, those are all elements or organelles that are found in these eukaryotic cells. So again, the planet arose about 4.6 billion years ago. Life arose on the planet about 3.8 billion years ago. Those first organisms were single-celled prokaryotes. And then eukaryotic organisms evolved on the planet about 2.7 billion years ago. And eukaryotic organisms evolved from prokaryotic organisms. And just so that you know, if we're talking about humans, you or I, our cells are eukaryotic, okay? Around 540 million years ago, life forms that you're probably more familiar with started to show up on the planet. So for example, you're probably familiar with arthropods, fish, land plants. How about mammals? We fit into that category. So mammals evolved about 200 million years ago. These started off as small mammals, and then more complex forms of mammals started to evolve over the years. We believe that human evolution began somewhere between 6 and 2.5 million years ago. And the fossil record, again, provides evidence for this, but we're unsure. That's a pretty wide time frame of exactly when human evolution started. About 300,000 years ago is when we start to see in the fossil record the evolution of humans that looked more like present-day modern humans. So if you do the math, it's pretty interesting. Organisms, humans, or human-like species, we've only been on the planet for about 0.04% of Earth's history, which is a small, tiny fraction of time in that big picture. That's pretty interesting. Okay, so in this chapter, these are the, this first part of the chapter, 1.1, section 1.1, these are the learning outcomes that I'm hoping that you take away from this. So you should be able to identify the shared characteristics of the natural sciences, and we'll go over what those are. You should be able to summarize the steps of the scientific method. We'll be using that scientific method throughout lab this semester. You're going to want to be able to understand and compare and contrast inductive and deductive reasoning. These are types of reasonings that scientists use. And lastly, you want to be able to describe the goals of basic and applied science. Okay. So first off, let's define biology. So the term biology means the scientific study of life. So bio means life, and logi means the study of. Okay, so it's the scientific study of life. Biologists will study living organisms. They may study single-celled organisms. They may study multicellular organisms. And biologists will also study how those organisms interact with each other and with the environment that they're found in. Okay, and so again, scientists, some will study even below the microscopic level. Maybe they'll study at the atomic level or at the molecular level or at the organelle level, maybe at the cellular level. They may study single-celled organisms. They may study multicellular organisms. They might study at the species level or the population level, the community level. So there's all different levels that scientists might study at. We're going to go over those levels in just a little bit. Pictured here on this slide, this is pretty neat. On the left-hand side, that image, this is right from your textbook as well. These are cyanobacteria. And so each one of these is a cyanobacterium. And so obviously we're using a microscope here. Cyanobacteria are single-celled, prokaryotic, microscopic organisms. So we're going to need a microscope to see those. And cyanobacteria are pretty neat. They are probably one of the first life forms found on the planet. If you look on the right, those are some of the structures in which cyanobacteria form. They actually form layers, and in those layers we find fossils. And we have fossils that date back to about 3.5 billion years ago. So right around when life started on the planet. These guys are still present on the planet today. They're really interesting. They're photosynthetic usually, sometimes even nitrogen fixing. Really neat to study. So again, some scientists will study at the microscopic level. Scientists who are studying cyanobacteria would fall into that category. So biologists, they study living organisms. I always like to put this slide in here. For those of you who are not biology majors, sometimes you wonder, why do I have to take this class? And we want you to be well-rounded. And from my viewpoint, every person, whether they're a biology major or not, every student needs to have some biological knowledge. You need to be biologically informed. Okay? Here's why. Prevention and cure of diseases. Oh, this is a really hot topic right now, right? Before we go to COVID-19, if I can draw your attention to this picture over here, these guys. So each one of these little rod-shaped structures in here is a single bacterium. And the bacteria that we're looking at in this particular image is E. coli. You have all heard of E. coli, right? There's some E. coli strains that are harmless. In fact, in the lab, we work with E. coli all the time. And then there are some strains of E. coli that are really, really nasty. This particular image of E. coli is taken from, it's actually right in your textbook, and this type of E. coli, this strain, causes some pretty serious intestinal upset. If you remember back, I'm not sure if we had any problems this past year, but last year, if you remember the E. coli on lettuce, lots and lots of lettuce recalls. It's been happening for a couple of years now. This is what the E. coli looks like that's on that lettuce. And if you eat that lettuce without cooking it, which you probably wouldn't, and without cleaning it really, really well, you may ingest some of this. And this E. coli causes some pretty serious stomach upset. Okay? You can treat it with an antibiotic because it is a bacterium. So it is treatable. However, people who get it, they get some discomfort, cramping, lots of things go along with it. And sometimes people will die from E. coli infections. Okay. How about the next one on that list, COVID-19? So hopefully you've been following this, right? And there's lots of scientific studies that are coming out on this virus. It's so new that the science, may change a little bit, and the general population will sometimes get upset with it. Well, you told me that I had to wash off all my groceries, and now you're telling me I don't have to wash off the groceries. You told me it could live on surfaces for days, and now you're telling me that it could only live on surfaces for like a day. Science is a slow process, and when scientific studies are rushed, sometimes there's misinformation. But in general, if you look at some of those studies, and I've read a lot of those, the science is good. At first, yes, COVID-19 can be found on surfaces for days. The key thing there is that it's not alive after a few days. Okay, so again, you have to kind of realize that this type of science usually takes years to produce results, and we're doing this in days, in weeks, in months. So prevention and cure of COVID-19, that should be on your radar, right? You don't want this. I don't want it. And if you get it, you want to make sure that you are going to be able to be treated for it. Okay, so finding therapeutics, finding pharmaceuticals to treat COVID-19, and then of course the vaccine for prevention should be really important to all of us. Okay, so we'll get away from viruses and from bacteria for now. Why else should you care? Why else do you need to be biologically informed? On the planet, there has been and there is still a huge decrease in biological diversity. Humans are creating the fastest ever species loss that the planet has ever seen. And it is human created. It's definitely not a natural process. We're speeding up what would be a natural process. And that's a problem. So a decrease in biological diversity is not good for the ecosystems on our planet. It's a serious problem that we should all be informed about and that we should all care about. What else? There's a decrease in natural resources. Things like clean water and fresh air, right? Those are natural resources that you should really care about. And with pollution, we're losing a lot of those natural resources. So that should be something that you care about. What else? An increase in greenhouse gas production. This is kind of a hot topic, right? You've all heard of climate change or global warming. Climate change is really the better term to use. this is something that you need to be concerned about. The planet is definitely warming. Sea levels are definitely rising. We're going to lose, parts of our coastal regions. There's going to be mass migrations of populations from that. And again, that's driven by humans. Yes, greenhouse gas production is a natural process, but we're producing much more than the planet can handle. So that's something that you should be aware of. You should also be aware that there's an increase in the human population. It's growing pretty fast, and that creates a biological problem. It creates stress on the planet. There's not enough food or clean water, natural resources, to take care of the whole human population. What else? So I just wanted to mention altering food production. There's so many more that I could add to this list. But this is kind of a hot topic too. GMOs, you've probably heard of those, genetically modified organisms. So when I say altering food production, that's what I mean. Taking food that grows naturally, and instead of doing selective breeding or artificial selection, now we're actually changing the genetics of organisms. So when you see that GMO, genetically modified organism, that means that in the lab we did something to the genetics of that organism to alter it. Maybe to make it so that it can grow in soil that's sandier, right, that has less water, or maybe so that it can grow in the presence of pests, okay, and not be harmed. The long-term effects of creating these GMOs right now is really not known, right, and there's a whole movement against GMOs, so it's kind of interesting. So that's just a short list of, some reasons of why you need to be biologically informed. Okay, and again, I could add to that list for days, but I'll stop there. Okay, so we defined biology, the scientific study of living organisms. And so just want to give you a couple of more definitions here that hopefully you have on that vocab list. So the next one is science. And when you hear the term science, science literally means to know. Okay, so knowledge. And that knowledge is going to be acquired, and it's going to be tested using the scientific method. Okay, and we're going to come back to the scientific method, the steps, in a little bit, but I wanted to just introduce you to what this method is. So when you hear that term scientific method, this is a research method that scientists will use that consists of a series of distinct steps. And one of those initial steps is going to be hypothesizing, and then that's going to be followed by experimentation. So let's go over what a hypothesis is. Okay, a hypothesis is going to be formed based on observation. Okay, so we start with observation, and from that observation, we draw a hypothesis. The hypothesis is going to be a tentative explanation or a potential answer to a specific question that we're asking, and that is what's going to be tested. Okay, and I'm going to be going over some examples of hypotheses with you in just a bit. Theory. So a theory in science is going to be something that has been tested and confirmed kind of over and over and over again. It's the highest rank of a hypothesis. It's based on a large body of scientific knowledge. Okay, so some examples of theories you've probably heard of: the Big Bang Theory and the theory of evolution, right? And so the theory is kind of the closest thing that we have to a fact or absolute truth. However, in science, as scientists, we understand that these theories, this is what we think based on huge amounts of evidence. However, if evidence were to come out tomorrow that disputes part of this theory, we're okay with that. Okay, so it's what we know today to be true. But if something comes out tomorrow that kind of changes that, we will then incorporate that new knowledge into our theory. Okay, we don't oftentimes use fact. We don't have that term because things can be changed. So nothing is 100% absolute in science. It's what we know based on the evidence that we have today. Okay, so that brings us to the natural sciences. So the natural sciences are going to be fields of science that are related to the physical world and natural phenomena. So there's a whole group of sciences that belong to the natural sciences, and biology is included in there. What else is included? Astronomy, chemistry, the earth sciences, and physics. Okay? All of those are included in the natural sciences. We could break those up into the life sciences and the physical sciences, but there's kind of a gray line with some of those. So the life sciences would include biology, and the physical sciences is kind of where you would lump everything else. However, you know, within biology, we study chemistry. Okay, so, and the laws of physics also apply to biological processes. So again, those lines kind of get a little bit gray sometimes. We say that all of STEM is interconnected. Okay, so you've heard STEM, right? Science, technology, engineering, and math. Some of those are natural sciences, some of those are life sciences, but all of those are related. Okay? Okay, so let's get into the types of scientific reasoning. And so our two types of scientific reasoning that we're going to cover are inductive reasoning and deductive reasoning. And I'll give you definitions for both, and then we'll give you some examples for both. So let's start off with inductive reasoning. This is going to be logical thinking that takes related observations, puts those together to arrive at a general conclusion. So I like to say it's going from specific results, specific individual results, and going to a big, broad general principle, going from specific to general. So let's look at an example. So I'm going to do inductive reasoning. I'm going to come up with a generalization based on two independent observations. Okay, so two related observations. I'm going to use both of those to draw a general conclusion. So here's observation number one. And this is, we're dealing with blood pressure and a specific drug that might be able to be used to treat blood pressure. So here's observation number one. So we've got scientists who did this study in rats, and they found that rats that are given drug X have lower blood pressure than control rats. Okay, so they had a group of rats. They split them into two separate groups. One group got drug X, the other group did not. They tested blood pressure, and the blood pressure in the rats given the drug was lower than the blood pressure of the control rats. Okay, there's one observation. Observation number two. So some separate scientists did some studies on dogs and cats. And it was found that if dogs and cats are given drug X, they too have lower blood pressure than control dogs and cats. So again, the control dogs and cats would be the ones that are not given drug X. Okay, so now we've got rats, dogs, and cats all given drug X, and they have lower blood pressure. So those are very specific results. Generalization. Here we go. So now this is the inductive reasoning part. Since drug X, right, works in rats, dogs, and cats, then drug X lowers blood pressure in all mammals. So do you see how we went from very specific results to, wow, we just went really broad. We're going to say that drug X works in all mammals. So rats, dogs, and cats are examples of mammals, but now we're saying it's going to work in all mammals. And that can be tested. Okay, we can test it in other mammalian groups. That's inductive reasoning, going from specific to general. The next one is deductive reasoning. This is also logical thinking. We're going to go in the opposite direction. It's going to use a general principle or law, a broad principle. And we're going to use that to deduce, to formulate, or predict specific results. So again, we're going from general now to specific. I'm not sure if you can see the bottom of this slide. Let me move myself up. Okay, so here is our example. If drug X is a safe and effective treatment for high blood pressure in mammals, very broad statement, then humans, now I'm getting specific, then humans administered 10 milligrams per day, that's really specific, of drug X will show a decrease in blood pressure. Okay, so I just went from general. Mammals, it works in mammals, it's going to work in humans. Now I can test that. Okay, here's some more examples from your textbook. And again, I'll move my camera image here. So again, this is again right from your textbook. It gives the definition for both inductive and deductive reasoning, and it gives you examples of both. So here's an example of inductive reasoning. We got a bunch of observations, right? Members of the same species are not all the same. Individuals compete for natural resources. Species are generally adapt to their environment. Those are a bunch of observations. And then we're going to draw a general conclusion. Individuals most adapted to their environment are more likely to survive and pass their traits to the next generation. By the way, that's the theory of evolution, right? Based on natural selection, right there. On the other side, on the right, we have deductive reasoning. We're going to go from general to specific. So the general premise here, individuals most adapted to their environment are more likely to survive and pass their traits onto the next generation. Okay, now we're going to get specific. So I can predict based on that general premise, if the average temperature in an ecosystem increases due to climate change, individuals better adapted to warmer temperatures will outcompete those that are not. Okay, I could test that. So again, I went from very, very general to specific, and those predicted results, that's my hypothesis really. I can go ahead and I can test that through a scientific study. Okay, so you want to make sure you understand the difference between inductive and deductive reasoning. Okay, next, there's two main pathways of science. Now we're going to talk about the two main types of science in a minute, but the two main pathways that scientists can choose to study. I want to make sure that before I go over each of these, even though there's two main pathways and it seems really clear and distinct, they're not mutually exclusive. So a lot of times scientists really do both of these pathways. Okay, so the first pathway is descriptive or discovery-based science. And scientists who do this type of research or follow this pathway of science, they usually use inductive reasoning, okay, where they go from specific to general. Scientists who do this type of science are going to aim to observe, explore, discover, to learn more about something, whatever it is that they're studying. And descriptive or discovery-based science tends to be qualitative in nature. Okay, they're gathering information, but there may not be a lot of numbers involved. Okay, scientists in this type oftentimes, are recording pictures or drawings or something like that, and maybe not actual numbers. The opposite of descriptive or discovery-based science is hypothesis-driven science or hypothesis-based science. And this is usually a deductive process. So again, you start with a general premise and you make a prediction or a hypothesis, and then you use the scientific method to carry that out and to test that hypothesis. And oftentimes, it's both qualitative and quantitative. It can also be one or the other, but oftentimes it's both. You're going to record observations, but you're also going to be recording and crunching numbers. Okay? And you're going to get some experience this semester in lab with some hypothesis-based science. Again, the two are not mutually exclusive. Sometimes science overlaps into both of those. Okay, so here is your first Are You With Me? So you want to pause our video here, and you want to have your in-class assessment handy, and you want to go ahead and you want to answer these questions. So you're going to start by defining some terms, and then you're going to compare inductive and deductive reasoning with some examples of each, and then you're going to compare descriptive versus hypothesis-driven science, and you want to compare qualitative and quantitative data. Now, before you start, I would pause the video, and I want to encourage you to not look back at your vocab list that you created, to not look back at your chapter outline, and to try to not even look back through your lecture notes that you've been taking. It's kind of like a test. Do you actually understand these terms? Do you understand the difference between inductive and deductive reasoning? Try to figure it out before you go and you look back. Okay, this is how you know, when I was doing my vocab list, was I just writing stuff down or was I actually learning it? When I was reading the chapter, did I actually understand it? As I'm sitting here listening to my instructor lecture about these things, am I understanding it? Okay, so if you can get to this part and you can start to write these definitions and answer these questions without looking back, then you're in pretty good shape. If you can't, then you know what you have to work on. Okay, so if you get to a point where you don't understand something and you do have to look back, that's okay. I want you to answer everything, so definitely look back. But you're kind of preparing your study guide, okay? And you're studying. There will be a quiz, right, at the end of all of this. Hopefully you'll take it towards the end of the week, and you want to perform pretty well. It's going to be a timed quiz, and so your time is limited, so you don't want to have to look back through your notes while you're taking that quiz. You kind of want to know the material. Okay, so go ahead and pause it, and when you're finished, come back and restart it, and I've got some questions for you. Okay, so welcome back. A couple of questions before we go on, okay, just to make sure that we're on the same page. So first off, the first forms of life on Earth were, okay, were they plants? Were they microorganisms, birds, or dinosaurs? So the first forms of life on Earth, what do you think? Give you a second to think about it. Okay, so hopefully you know that the first forms of life on Earth were microorganisms. Okay, organisms that are single-celled that you would need a microscope to see. The second, which of the following sciences is not considered a natural science? Is it biology, astronomy, physics, or computer science? Which one of these does not deal with the natural world, so the natural sciences? I'll give you a minute. Okay, so the one from this list that is not a natural science is computer science. Okay, definitely part of STEM and related to the natural sciences, but not one of the natural sciences. Here's another question. All flying birds and insects have wings. Birds and insects flap their wings as they move through the air. Therefore, wings enable flight. Is this an example of inductive reasoning or deductive reasoning? Okay, so we went from some specific things, right? Birds and insects both have wings. Birds and insects flap their wings as they move. And so then we went to a really general statement. Wings enable flight. Okay, so that's an example of inductive reasoning, going from specific to general. The second question here, insects generally survive mild winters better than harsh ones. Therefore, insect pests will become more problematic if global temperatures increase. So is that an example of inductive reasoning or deductive reasoning? I'll give you a second to think about that. Okay, so we went from general to specific. We could test that, right? If temperatures increase, do pests become more problematic? That's going from general to specific, so that's an example of deductive reasoning. Okay, a couple more. So the first one here, Jane Goodall, she's a pretty famous scientist who studied chimpanzees. She would record qualitative observations of their behavior. So she would draw pictures and write about their behavior, how they interacted with each other. Her efforts helped to raise awareness of these animals both in the wild and in captivity. How would you classify her work? Was it descriptive and discovery-based science, or was it hypothesis-driven science? I'll give you a second to think about that. So hopefully you answered, that's an example of descriptive or discovery-based science, right? Qualitative in nature. Yes, she was recording observations. Probably didn't use the scientific method, wasn't really testing anything. More was observing their behavior. Move my camera up here. Second question here. Researchers are carrying out carefully planned experiments using xenobots. Actually, this question I pulled out of something that I recently read about. So these things called xenobots, researchers are using them. They're driving carefully planned experiments. This new technology is being studied for use in things like radioactive waste cleanup, collection of microplastics in our oceans, carrying medicine inside the human body, and maybe they can even be used to scrape out plaque from the insides of human arteries. Scientists are collecting both quantitative and qualitative data on these little xenobots. How would you classify this work? Is it descriptive or discovery-based science, or is it hypothesis-driven science? I'll give you a second to think about that. Okay, this would be hypothesis-driven science, and you would know that based on the very first sentence there, that researchers are carrying out carefully planned experiments, right? And they're also gathering both qualitative and quantitative data. Okay, so a little bit more, still in chapter 1 in that first section, we're going to move on to talking about the scientific method. Let me move my camera down here so you can see that picture. Hopefully. Okay, so the scientific method, this has been used for a very long time. It's been used since ancient times. It was first documented by a scientist by the name of Sir Francis Bacon. He lived from 1561 to 1626, so it was during that time frame that the scientific method was first documented. But again, it had been used prior to that. The scientific method, he used it to set up carefully planned experiments that he would then carry out. And we still use that today in biology and in other scientific disciplines as a logical problem-solving method. So we're going to go over those steps. Okay, so in the scientific method, you're going to start with observing. Okay, oftentimes what you're observing is some type of a problem that needs to be solved. And from those basic first initial observations, you're going to come up with some questions. Okay, here's an example. And I'm going to go back to the drug studies that we talked about before. So here's an observation. A certain percentage of the human population suffers from high blood pressure. Okay, from that observation, this is a question. Could drug X be an effective treatment for high blood pressure in humans? Okay, and oftentimes we'll associate with that other observation. So if there's drug trials that have been done in other species, we would add that to the observation. Okay, so here's the hypothesis. By administering drug X at 10 milligrams per day, that will safely and effectively lower blood pressure in humans that have high blood pressure. So that's the beginning. We make an observation. From that observation, we formulate a question, and then from that question, we formulate a hypothesis, a potential answer to that question. Okay, and it's something that can be tested. Okay, now, after we formulate the hypothesis, we then will conduct an experiment. And I'll move this out of the way. Okay, so we'll test that. We'll test that hypothesis. It's key when designing an experiment that you only test one variable at a time, and you always want to make sure that your experiment is controlled. Okay, so let's look here at our example of this drug study. So at the very top of this figure right here, we start with our sample, right? So we have a large number of humans in this case, and all of those humans would have high blood pressure. Okay, and then we're going to take that group of humans and we're going to randomly divide them into two groups, right? And we're not going to use things like age or weight or gender or ethnicity when we're breaking those two groups up. We're going to do it randomly, okay? Again, all of them have high blood pressure. Now you've created two groups. All of the humans in both groups have high blood pressure. And you're going to take group 1 and you are going to give them the treatment, right? So they're going to be given drug X at 10 milligrams per day. The second group, group 2, is given a placebo, okay? And you probably know what that means. They're still given something that they think might be drug X, but it's not actually drug X. It's usually just a sugar pill, okay? And we could make this what's called a double-blind study, which is even better. And the doctor or the nurse or whoever the medical professional is that's administering this medication and then looking at the results, they wouldn't know either whether their patients are getting the actual treatment or if they're getting the placebo. Okay, and that goes for both groups. Group 1 is going to get the drug, but they know that they might actually be getting the placebo. Okay, so you've got your experimental group, you have your control group. The only variable, the only thing different between the two is whether or not they get drug X. So after you kind of set this up, you then are going to gather your data and draw conclusions, and this is going to be quantitative data. So blood pressures will be taken of both the experimental group, group 1, as well as the control group, group 2, and then you're going to compare the results. So what happened? Are blood pressures lower in the experimental group compared to the control? If they are, then you would accept the hypothesis, right? That hypothesis would receive support. Of course, these studies would have to be repeated. If not, then there's a problem, right? And so you'd have to go back and look at that hypothesis again and alter it. Okay, maybe the drug dose wasn't high enough, or maybe the study didn't take place long enough. Okay, so again, you have to use the scientific method to carefully plan out and execute the scientific study. Okay, here it is in a pictorial form for you. So again, at the very top, you're going to make an observation. Based on that observation or observations, you're going to ask a question. From that question, you are going to form a hypothesis. You are then going to test that hypothesis, analyze your results, and either accept or reject the hypothesis. Okay? And of course, you're going to have to report your results. And we'll talk about how those results are reported in just a little bit within this chapter. It seems like everything is very methodical, right? Every step of the way is carefully planned. I want to make sure that you understand the role of serendipity. And I think your textbook talks about this as well. Serendipity is a fortunate accident. And this definitely plays a role in science and in biology. A great example of the role of serendipity is with Alexander Fleming and the discovery of penicillin, right? Penicillin you guys probably have all heard of. It's an antibiotic used to treat many bacterial infections. We actually found out what penicillin was and its capability of inhibiting bacterial growth on accident. Okay, so Alexander Fleming was a scientist. He was growing bacteria on a plate, and that bacteria, the plate became colonized with a fungus, right, on accident, right? He didn't want the fungus on his plates, and luckily, instead of just tossing out his plates and starting over, Alexander Fleming took a closer look, and what he found was where the fungus was growing, there was this big ring around it where there was no bacterial growth, and so he figured out that that fungus must be producing something that inhibits bacterial growth, and that something was penicillin. Okay, we wouldn't have found that if he just threw his plates away. So yes, there's definitely a role of serendipity in science. Okay, so this next slide, we're just going to cover the two main types of science, and they're kind of related to the two main pathways of science as well. So the two main types of science, we've got science that is basic science, or we can also call it pure science. Scientists that do this type of research they're going to be doing their research mainly to seek knowledge, purely to gain insight into how a species lives, or how a species survives, or something like that. The findings may not be practically applicable, or they may be. But that's not the purpose of basic or pure science. It's just really to learn more about something, some organism. Okay? Applied science is science that is used to solve real-world problems. And again, there's a gray line between the two of those, because a lot of times science starts off as basic science and then becomes applied science. So for example, I've got a couple of images of two different species here on the right. Right from your textbook, the first one is, I'm not sure about the second one, I think that was from a previous textbook. So at the top there you see that little tiny squirrel. So years and years kind of have gone by, and scientists have studied squirrels, okay, basically to learn about how they live, what their nesting is like, what they eat, and that would be just basic pure science. But after Hurricane, I think it was Katrina, yeah, I think so, Hurricane Irma, that basic or pure science turned into applied science. So a lot of these squirrels had fallen from nests. They were baby squirrels that were abandoned by parents, and so there was this need to use that basic pure science that had been gathered over the years to help these organisms to survive, to kind of save the species. Since we knew about nesting and we knew about how they ate, we were able to rehabilitate a lot of these squirrels and put them back out into the wild. The same thing at the bottom there with that pelican. This one's from Hurricane Katrina. So during Hurricane Katrina, a lot of pelican habitats were destroyed. And so because we knew a lot about pelicans, how they lived, how they nested, how they mated, what they feed upon, all of that, we were able to, again, rehabilitate and put back into the wild a lot of those pelicans that were left kind of stranded without homes and disoriented after that hurricane hit. Okay, so again, science a lot of times starts off as basic pure science, just seeking knowledge, but it will turn into applied science in the future. So they're definitely intertwined. Here's another example. Again, started off as basic pure science and is now being used as applied science. So the Human Genome Project, you've probably heard of it. It was a 13-year collaborative effort between researchers from all over the globe. It started off as just basic pure science. We wanted to know the genome of humans, right? And we've done this for other organisms as well. We wanted to know what was the code, what was the order of the bases, the letters in our DNA, okay? And so we figured that out in the early 2000s. That project was complete. And now we know the human genome, right? And there's definitely, not every human, not all our DNA is not 100% identical, but a lot of our genome is. And so that's the basic pure side of that. And now it's being applied. So now we can take, our genome and we can figure out disease susceptibility based on what form of a gene that we have. We're figuring out what genes code for, what those protein products are. And we're actually even starting to alter genes, right? We're doing genetic modification. We're using CRISPR technology to change genetics to decrease things like disease susceptibility. Okay, so again, started off as basic pure science, and now it's becoming applied science, and it's really helping the human condition. Okay, so here is your second, are you with me? So again, you're going to pause your video here, and you're going to go through and you're going to answer these. So again, you're going to describe the steps of the scientific method. So I would list those steps, and I would define each of those steps. And then the next part's kind of cool. I want you to think about and design your own research question based on an observation, and you're going to use the scientific method to address it. What would you do, hypothetically? Make up some data that you've gathered Either accept or reject your initial hypothesis. Again, this is just hypothetical, so it could really be anything that you're interested in. So go ahead and just give that a shot. And that second question there, what is the difference between basic or pure science and the applied sciences? And the second question, again, I want you to think about it. What are your thoughts about public funding for each? Okay, most scientific research that takes place is publicly funded through, for example, the NIH, the National Institutes of Health, which is a government agency which takes taxpayer dollars and funds scientific research. What do you think about that? Should yours and mine, should our tax dollars go to support basic pure science, just learning about things for the sake of learning about them, even if it's not applicable? Or should our tax dollars be going towards just applied sciences? Kind of want to know your thoughts about that. Okay, so go ahead and answer those, and then turn me back on when you're finished. Okay, so here's just a couple of questions for you. I think we got, a few. Okay, so the first question, in designing an experiment to test the effect of music on plant growth, the scientist exposes the first group of plants to music and does not expose the second group. This second group is treated the same as the first except the exposure to music. The second group is called is it the variable, the experimental group, the control group, or the hypothetical group? So hopefully you said the control group. Second question for you. A suggested and testable explanation to a scientific problem is called is it a hypothesis, a variable, a theory, or a control? So hopefully you said hypothesis. A couple more. These are examples that we actually already saw. So Jane Goodall's studies of chimpanzees. Again, she would record qualitative observations of their behavior, and her efforts did help raise awareness for these animals in both the wild and captivity. Her work would be classified as, this is kind of a trick question. So initially I would say her work was just basic or pure science. She was just learning about chimpanzees. But then the part where she raises awareness for animals and maybe how they were treated, both in the wild and in captivity, that's definitely applied science. So I would say that there's kind of a fuzzy gray line here. I would kind of classify that as both. Okay, second question. The researchers that are carrying out experiments with these xenobots, right? They're going to be used to do lots of things that are going to help the human condition. They're going to collect both qualitative and quantitative data. Would you classify that as basic or pure science or applied science? Definitely applied science, right? We're definitely going to be using these xenobots to help the human condition. We're going to apply what we're learning from them. We're not just gathering data to gather data. We're going to use it. Okay, so before I go on, what I'm going to do is I'm going to stop this video here because we're at almost an hour, and I'm going to create my second video, okay, just to kind of break this up for you. So I'm going to pause this here, and if you want to keep going today, you're going to just open up that second video, or you can watch that second video at a different time, just to break it up a little bit.